Hongyi. If I can give you a quick introduction, he did his PhD at the University of Queensland finishing in 2013, specializing in electron microscopy and semiconductor nanomaterials. In 2014, Hongyi started a postdoc fellowship in the lab of Professor Xiaodong Zhu at Stockholm University, and there he's initiated the development of micro-ED, which is what he'll talk about today. He became a principal investigator in 2018 and is continuing to develop and apply electron crystallography methods for studying structures of biomolecules. So if you're ready, would you like to start?
Thank you very much. So first of all, I would like to thank the organizers for giving me the opportunity to share what we've done in recent years in terms of the development of 3D ED and micro-ED. And also a big thank you to all those people who put tremendous efforts in making this symposium possible in this very difficult time. So today I'm going to share to you about with you about what we've done in the field of 3D ED and micro-ED, which are 3D electron diffraction methods for solving structure from tiny crystals. Okay, so why do we use electron to do crystallography? Because 3D ED and micro-ED is complementary to existing techniques. And a lot of the X-ray softwares that has been well developed in the past years can be directly used to treat electron diffraction data. And this has helped us to develop this method very fast and very rapidly. And one of the major reasons that we use electron instead of X-ray is that it interacts with matters much stronger. And so in terms of the crystal size, we can now work with very tiny crystals. So those crystals that are too small to be studied with X-ray can now be studied with electron diffraction. So if this is the size of the X-ray crystal, this is the crystal you need to do electron diffraction. So this is just a comparison. And I've stolen this picture from a postdoc, and I think it's very good. So normally you set up a crystallization drop. You're looking for these very nice large crystals. But most of the time I think you also see these very thin needle crystals. Actually, these thin crystals are perfect for micro-ED experiment. Okay, so electron also interacts not only with nuclei, but also with electron clouds. So it is possible to see charge in the electrostatic potential map generated by electron diffraction data. And also because electron interacts with matter so strong, so we get dynamical effect from electron diffraction. So this, using the dynamical refinement, we can actually study the absolute structure of chiral crystals. In this case, that Lucas Palatinos' group last year showed that it was possible to treat the dynamical scattering in electron diffraction data in order to solve a absolute structure. And moreover, as I think the cryo-EM community knows, that electron can be focused, and we can get high resolution images, not only from single particles, but also from crystals. So this may one day help us to phase our diffraction data. So the phase information is contained in high resolution images. And this may be a new way to do de novo phasing from electron diffraction data. Okay, so there are several ways of collecting 3D micro-ED data. The most popular way by far nowadays is the continuous rotation data collection. The strategy is very simple. All we have to do is to rotate the crystal inside of a TEM continuously while we're taking very fast frames in a movie mode. We just collected the diffraction frames continuously, which is very similar to what the people are doing for synchrotron nowadays. And after we have collected data, we can reconstruct it and put it into, reconstruct the 3D reciprocal lattice and then extract the intensities from indexed peaks. And this can be done with our in-house software RED-P and also with standard X-ray software. And most of the X-ray software works really well with the electron diffraction data. Okay, so with 3D ED, micro-ED in Xiaodong Zhou's lab, we've solved more than 200 structures in the past 7-8 years. And as you can see that the lab started working on material science. So we've been looking at the novel complexes of zeolites, COFs, germinates, quasi-crystals, and also metal-organic frameworks. And I joined the group in 2014, and in late 2014, I started to look at the possibilities of applying 3D electron diffraction techniques to organic molecules as well as proteins. Okay, so I've tried to put everybody who's working in the field on the map. I mean, I apologize if I've missed anybody, but the field is very small and it's very vibrant, so I can fit everybody on the map. And as you can see, there are people doing this in Europe, also in Asia, in Australia, also in the US, where the first micro-ED work started in Tamir Gunan's lab. And as you've heard from Ming Song yesterday, that EBIC is almost ready to accept samples from users to collect micro-ED data on. And the first protein structure by micro-ED was solved in 2013 by Tamir Gunan's lab. And up to May 2019, there have been 32 entries in PDB of 13 different proteins. And but all these proteins are previously solved by X-ray. So the phasing was done with 100% sequence identity homologs. And there was only a new polymorph of lysozyme that was discovered by Maura Jemi in 2019. And the first unknown structure was solved by our group in 2019, last year. Okay, so after the introduction, I would like to give you an outline of my talk today. So first, I will cover the method development of micro-ED. So what we've been doing to make the method more accessible, more reliable, and more user-friendly. So I'll start by talking about sample preparation, which is a method we developed to work with micro-ED specimens. And we call this pre-assist, pressure-assisted method. And apart from this, we've also been working a lot of software on the software side. So we've developed methods called platforms called Instamatic, which allows people to collect 3D electron diffraction data rapidly. And also we have Instamatic, which is a DM version of Instamatic. So I'll talk about this in more details later. And also I'll talk about how we treat the data now, because we're collecting data so fast nowadays, and we need to have a pipeline to process the data. And after that, I'll talk about some of the applications we've been using 3D ED and micro-ED to solve structures. So I'll talk about the current workflow at our lab, and I'll show several examples. And then I will be wrapping up with the future perspectives. Okay, so as we all know that sample preparation is probably still one of the bottleneck in cryo-EM, not only single particle, but also it's for micro-ED. Because a lot of the crystals people nowadays grow are growing very highly viscous solution. So it is very difficult actually to remove this viscous liquid while keep the crystal hydrated and attached it to the grid. So we developed a very simple method. It's just putting a piece of filter paper on a flask and then rest the grid on top of this filter paper. And then before we apply the sample, we literally bring a suction underneath the sample. So when we drop the liquid containing the sample onto the grid, the liquid is sucked through the to the filter paper, and then the crystal hopefully sits nicely on the grid and still hydrated. And then we manually lift this grid and then plunge it into liquid ethane for freezing. And if we compare this with Vitrobot, so this particular solution here is growing 44% of PAC 400. We've been trying a lot of Vitrobot conditions using double filter papers, double blotting, but this is the best we could get, maybe 1 or 2 holes in the entire grid. And even if we have a hole, the ice is way too thick. And if you look at the pre-assist case, that we get very nice grid, and we can find many crystals on the grid. And because of this is blotless methods, we end up with much more crystal on the grid as well. Okay, so we can also tune the ice thickness with this method, and there are 2 major parameters. One is the pressure we apply to the underneath the filter paper, and also the second is the hole size of our quantifoil grid. So it's very logical, right? So if we have very large holes with large pressure, we end up with a dehydrated crystal in this case. And then the ice is the thinnest. And if we look at the other extreme case where we have very small holes, but small suction underneath it, we get ice rings. So the ice is slightly too thick. So for micro-ED experiments, actually, we want to have the crystal the ice around the crystal as thin as possible while keeping it hydrated. So in these two cases where we have small holes and large pressure, or large holes with smaller pressure, we can get very nice grids, and you can see the resolution extended to much higher than in the other two cases. Okay, so we apply this to other systems as well. In this case, the DNA cytochrome crystal, almost 2D crystal, we can have very thin conditions where the crystal is almost dehydrated. And we have some intermediate case where some holes are empty, but some holes are still wet. And we can have very thick ice as well. So this is really tunable for different systems. And of course, we've been using this method in a lot of our experiments. And as you can see in these cases, just from a small portion of a grid square, we can get many, many crystals that is suitable for micro-ED data collection. So each crystal, we can get one set of micro-ED data. And most interestingly, we've been also working with Team Grunen's group that on this 3D support, because when we have plate-like or needle-like crystal, we have a preferred orientation problem on the grid. So what they thought about is to bring nylon fibers onto the grid. So we have a 3D support. And we worked with it, and it really works very well with our methods. We can have like a 3D support for micro-ED. Okay, so we also extended this method to a single particle because it worked so well with micro-ED. So we thought, why not give it a try with single particle sample? So the idea is very simple. We built a add-on to the Vitribot, and so all we have to do is to deposit the sample as it used to be, and then instead of blotting, we bring the tube to actually suck the liquid through the grid and leave a very thin layer of liquid on the grid, and then just plunge it into the ethane without blotting. And this has actually received quite a lot of attention, and I think a lot of constructive criticisms as well. So we've been working very hard on improving this method, and we still believe that it works quite well. So this is the latest iteration of this method. So we now have a much better tube. It has been modified, and now the position of the tube can be reproducible every time. And by doing this, I mean, this is from last week, and we can get quite consistent ice thickness on grid. Unless in this case is not very nice, but all the other 4 cases we get usable areas. And of course, we could reconstruct the epiferritin structure to 2.6 angstrom resolution. So we still believe in this method, and we're still working very hard on it. And hopefully, in the near future, can be brought to the community. And the major advantage of this method is that without this blotting process, we can keep a lot of sample on the grid. So we can work with sample with very low concentration, as low as 0.18 milligram per milliliter for epiferritin. And there are other things we can play with the method as well, I believe. Okay, so that's enough for sample preparation. And now it's for data collection. This is a Python-based system that is developed in our group by Steph Smith, a postdoc who left our group, unfortunately, end of last year. But the system is very, very powerful. So it talks with the microscope, in our case, a geo-microscope. And also it talks to our detector, which is a Timepics detector. And it can allow people to implement different methods based on Python libraries. So currently we have serial ED and CRED data collection, and also we have automated CRED data collection nowadays. But there's a problem because this software, we cannot get APIs from Gaten cameras. So this software, we can hack it to talk to Thermo Fisher microscopes now. So it's compatible with most TEMs, but not all detectors. And we are working on more hacks to make it happen in the near future. So this is what the system looks like. So we have a live view of the detector, and we have our exposure time. And here is is image interval. I'll talk about this during the video. And as you can see, we have different methods implemented into this software. So this is really extendable, and it can take the advantage of the Python library. So that's how the data collection looks like. So first, we defocus the electron diffraction beam to form an image. So this is in diffraction mode and continuous rotation. And during the rotation, we have this image interval settings. So this is saying that after 10 images, we defocus the diffraction beam to form an image. But during this image formation, the ganometer is not stopped. So we are still rotating, but to form this image, it helps us to relocate where the crystals are. So when we continue to rotate the crystal moves, but we have a way to form an image. Then we can use this image to put this crystal back into our diffraction beam. So it's very handy. And so it goes like this for about 2 to 3 minutes for inorganic sample in this case. And at the end of the data collection, you can see the crystal shape changed from rectangular to some thin plate because of the rotation. And then at the end of this data collection, we can actually process the data online. So in this case, I'm showing you the XDS add-on we put on the microscope computer. We have to run this in the VirtualBox because it's not Windows 10. But as you can see, the data is collected, and we can transfer it into the Linux VirtualBox, and then XDS can be run to process the data live, which is quite nice. Okay, so as I talked about before, that Instamatic works with almost all microscopes now, but not all detectors. And the majority of the lab are actually using GATAN detectors. So we came up with the idea that maybe it's very useful to develop a DM script that can control the data collection to allow micro-ED data collection. So the name of the script is called Instamatic. I think it's brilliant naming by Steph, DM-matic, right? So it's a simple DM script which allow you to continuously grab images from GATAN detector, while it can send also commands to the FVR microscope to tell it to rotate. In this case, actually on our system, our computer to control the microscope is different to the computer that controls the GATAN detector. So we have two computers which we have to go back and forth. So we use IP port to do this, to send commands. But it just shows that it is now possible to use the DM script to collect the micro-ED data with GATAN detector. And the manuscript is submitted, and you can see the preprint. Okay, so that's after we collecting so many data sets in a very short amount of time, the data processing becomes a problem. Right? So we need to have an automated pipeline to allow us to process the data fast enough. So what we did was we teamed up Dials and Sypian. The Dials is a software developed at Diamond Light Source to process X-ray diffraction data. And the name is very good. It's diffraction integration for advanced light source. I think Electron is the advanced light source as well, I think, in the future or already now. And Sypian is actually a management software that allows to process single particle data in a pipeline. So a lot of labs are already using Sypian. And Jose is going to give a talk about Sypian tomorrow, I believe. And so what we did is Victor implemented Dials into Sypian so that you can have a flow chart of the Dials process. And you can use a very easy to edit dialog window to input all the inputs for the micro-ED experiments. And we believe in the near future, we can also set up the streaming process like what a lot of single particle labs are doing. So the system can monitor the incoming data, and once the data set is collected, we can process it automatically in the pipeline. And this software will be available very soon. Okay, so now we have so many data sets processed, and how do we merge them? I mean, for micro-ED data, because the beam sensitivity of the crystals, we have to actually merge a lot of data sets. So one thing Xiaodong came up with this hierarchical cluster analysis. So this is doing 2 clustering processes. So the first one is to look at the unicell dimensions of each data set collected from different crystals. We cluster them based on unicell, and then the second clustering is done on the CC, which is the correlation coefficients between the data sets. And by doing this, we can just use Instamatic to automatically collect data and then process all this data, cluster all this data afterwards, and we can distinguish different structures. In this case, zeolites and modernite, those materials. It's doable with this clustering process. And yeah, so in terms of protein, we actually need to do this merging, as I mentioned before, because in this work, we've shown that by merging lots of data sets, maybe because of the preferred orientation, by merging lots of data sets, we don't get so much higher completeness in this case. So 1 crystal give you 47%, 4 crystals 75%, 33 crystal reached almost 80%. But you can see if we see the multiplicity, which is the redundancy, in other words, we get a very high redundancy in 33 crystal data set. This can enhance our data quality and also the final model, the quality of the map. As you can see in the 33 crystal case, we can see the sodium ion in the lysozyme case. And by looking at the omit map, we get a very nice map with the 33 crystals. So merging data is very useful. And nowadays we are doing a routine that we collect them maybe 15, 20 degree data from each crystal, and we can collect so many crystals, and then we merge them and sort them out using the clustering afterwards. Okay, so that's about enough about the method development we are doing, and now I'm going to show you some of the examples and the workflow we've been having in our lab. So nowadays we are not biologists, so we are working closely with structural biologists who grow crystals. So there are 2 methods of shipping samples now. One is to ship the crystals in Eppendorf tube, already grown crystals in solution, and also people can also ship in protein solutions. And we are setting up a dedicated crystallization bench for crystallizing our lab before the experiment. And later on, I'm also thinking about studying how to optimize these crystals for micro-ED, de-optimize actually of these crystals to make them small enough for micro-ED experiments. And for specimen preparation, we have this pre-assist method, and now it takes about 2 to 3 trials to get a very nice grid. That's something I'm really proud of, and the reproducibility is really high. So this allows us to work in a very high throughput manner to screen different samples. And then we go on with data collection and analysis and structure determination. So if the crystal diffract really well, these processes takes only 2 days. And this has happened for many inorganic sample and the small organic molecule sample, and also happened to 2 protein crystal samples up to now. But there's a problem that 70% of the crystal we receive that does not diffract. And they look like crystals, but they just don't diffract. I also heard this is very common for X-ray case. So that our current screening throughput is about 4 to 5 crystal forms per day. And our experience tells us that plate-like or needle-like crystals are more likely to diffract. So the mentality is slightly different to X-ray. We don't look for one single large crystals, but we look for many crystals of the same shape. And this is telling us that there's a high probability of they are packing the same way and they're probably crystal. And we're also interested in developing new screening methods. Okay, so another very good thing about our lab is that the methods developed here in our lab are directly transferred to the Swedish National Cryo-EM facility at SciLife Lab. And thanks to Matthew and Marta, we are already accepting micro-ED samples from users, I think all over the world. The last time I talked to Marta yesterday, and she said, we are happy to look at people's sample now at SciLife. Okay, so next I'm going to show you some of the examples we've done by using micro-ED. So first is the SAR2 Lux, was the first protein solved, unknown protein solved with micro-ED. So we phased it from a 35% homolog with 35% sequence identity. So this is the very viscous sample I've shown you with the sample preparation. And one thing I want to point out, the resolution 3 angstrom, not very high, it's reasonable. And completeness is 63%, which could be a problem for the future because the crystals are all plate. They all sit on the grid in the same way. And if you look at the map, we didn't actually fill any data during Phoenix refinement. So this is the raw, I mean, this is the refined map from only 63% data. It's not so nice, but it's quite reasonable, I think, for 3 angstrom resolution. Okay, so the next thing I'll talk about is MyD88. It's a signaling protein sitting underneath toll-like receptors. And these crystals are actually naturally formed. So they're almost like filament like crystals. And these are the crystals, what they look like. And when we put it into the microscope, it's very interesting because these are very thin needle-shaped crystals. So they like to bundle together, right? So, so if we have a huge bundle here, in this case, we get very blurred diffraction spots because of multiple crystals. And if we look at if this is like 2 or 3 crystals, we get almost a single crystal diffraction now. And if we find a single crystal, we can get beautiful diffraction patterns, and we can collect the 3D data from them. And this actually, our collaborator actually put this on the XFEL beamline as well. But because of this bundling problem, they couldn't get very nice high resolution data to solve the structure. And again, this structure was very different to the crystal structure. So this naturally formed crystal resembles more to the natural case in the body. It's not a crystal, it's not different to the crystal structure. We actually have to face it from 30% sequence identity homolog. Okay, next I'll talk about Isot, which is a software we used to refine this.
Can you wrap up in a couple of minutes?
Sure, yeah, sure, yeah, sorry. So this is very useful software for low resolution data. I think I'll skip. So the important thing is that you can see this refinement live. If you drag it off the map, it refines back into the map, right? It's a live view. It's very useful for micro-3D data. And next I'll talk about how much fine details we can see in the structure now. So this image is showing you the difference-free map imposed with the structure. So you can see all the water and even hydrogen atoms in hydrogen bonds can be refined directly from micro-3D data. So it's very, very high quality now for micro-3D data. And in this case, we have a MOF that we can see the organic molecules. So this gives us a question, can we see this in drug binding? So that's what we did recently. So we have a HCA2 protein, and then we bind the drug to it. And if you look at the map, this is native, where you have the zinc atom in the difference-free map. In this case, apart from the zinc, you have extra density, which fits nicely to where the molecule should be. And if we compare with other techniques, we have quite a nice map. I think it's slightly better than neutron, and this neutron map is jointly refined with X-ray data, but X-ray data still has the best quality. But micro-3D, we show that it's capable to refine drug binding in the protein structure. So next I'll skip probably. This is the crystallization on the grid. So this is a small molecule we crystallized on the grid. With different time, we can get different polymorphs of this of this molecule. And in the future, I think it'd probably be possible to grow protein crystals on the grid directly. Okay, so hopefully I've convinced you that micro-3D is a very useful method, and it sits in this map here. So if you have small molecules, small crystals of small molecules, it's perfect for micro-3D experiments. So probably start thinking about this method and give it a try. Okay, so finally I would wrap up with ongoing projects and outlooks. So we are working on more to improve the sample preparation and to do high throughput experiments and make it easy for everybody to use. And from yesterday's talk, hardware improvement is also very important. We also dream of an electron diffractometer dedicated for doing micro-3D. And something I've been working on, just a small tease, if you can see this rotation of this grid, actually this time it can rotate more than 180 degrees in the TM. And the beautiful thing about this system is that it rotates so well that the drift is minimum. So you have minimum drift with very high tilt range. So that's something I've been working on very hard, and hopefully we can bring this to everybody in the near future. And also we are working on experimental phasing and time-resolved crystallography. Okay, so this is the whole group. We are a very large group, and I'm leading a small group of 6 people that working on very hard on developing micro-3D for protein, to solving protein structures. And we are hiring PhD students and postdocs all the time. So if you're interested, email Sheldon or me. And last but not least, we have a workshop that is adjacent to the IUCR meeting now next year in August. Thank you very much.
Okay, thanks very much. We have a lot of questions. I'm going to try and group some of them. So there are a couple of questions on your non-blotting sample preparation technique. So Emma Stepinak, would pre-assist preparation work for oil-rich conditions as well? And then we also have Cécile Petit, basically asking if you have problems with crystals cracking using this technique. So maybe we can put this together.
Yes, yeah, we haven't worked on any sample that has been oil conditioned, but it will be very interesting to try. I think it should work. I mean, we are working now with LCP crystals and or lipid sponge phase, at least. And we are also looking at the possibilities to work with mitochondria samples, and it works very well. And we can have mitochondria, and we can have a lot of beads in the host as well. So yeah, it will be very interesting to try oil samples. And the second question about crystal cracking, we haven't seen any crystal cracking yet, I think. The reason why is that I think crystals actually like to bring liquid with it. So it's actually very difficult to dehydrate a crystal sample using this kind of method. And we haven't seen any crystal breaking. And that brings me to another thing that I forgot to point out is that for ligand binding, I think a smaller crystal has less chance to crack compared to large crystals as well. So with microcrystals, we haven't seen anything cracking. And even if they crack, we can probably put a smaller beam on it and collect data on it. So small is not a problem.
Okay. So yes, in fact, there were several questions about the optimum crystal size.
Yes.
And I think probably the most direct one was Elizabeth McLean asking if you, I'm just trying to find it, don't want to misrepresent her. Do you crush larger crystals to get small ones?
We tried this after Tamir's Gonen's group published this fragmentation method, but it wasn't very successful. So at the moment, I think for crushing crystals, we can use ultrasonication for a very short amount of time. We can use beads, like a seeding procedure. But I think after using the seeding procedure, it's necessary to take some time to actually for the crystals to recover. So what we found was when we crack crystal, the resolution dramatically was dramatically lowered. For example, if it diffracts to 1.8 angstrom in X-ray, when we crack the large crystal for micro-ED, it gets to about 4 angstrom resolution. So there is the effect in reducing the crystallinity. But I think different system also behaves differently, and it will be very interesting to try different system and see how cracking would affect the crystals.
Okay, thank you. So I think final live question, Jacob Brink. How do you reposition the crystal in defocused diffraction? So I think that's what you're showing that every 10 images.
Yeah. Okay, so for for protein data set, we actually don't do this process. We collect around 15 to 20 degree of a small wedge. And if we align the microscope properly, it shouldn't move without, away from the beam. But for this repositioning we are doing is that when we defocus the reflection, we at least get an image of where the crystal is in relation to the beam. So during the next 10 frames, we know how to move the crystal back into the beam. So it's like looking at snapshots of the movement and try to compensate it using the trackball. And before this was done, actually, we were able to blindly track crystals with experience. But now with the images, I think it's much easier to track the crystal.
Okay, that's great. Thank you. So I think we'll take the other questions offline. So thanks very much, Hongyi.
Okay, thanks.